In this lecture, I will be talking on allergic bronchial asthma or atopic asthma. Asthmatic responses lead to degranulation of mast cells, and as can be seen here, this is the mast cell. They are tissue cells. They express Fc epsilon R1 high affinity receptors specific for IgE antibodies or IgE raised against allergens. In allergic individuals, nearly 90% of Fc epsilon R1 or these high affinity receptors are occupied by IgE to allergens, and this takes place during first exposure. In second exposure, the allergen, as can be seen here, this is the allergen cross-link the bound IgE antibodies, and this cross-linking of membrane bound IgE antibodies will send two signals. One signal to the vesicles and lead to degranulation of the vesicles with liberation or expression of histamine, platelet activating factor, eosinophil chemoattractant factor, neutrophil chemoattractant factor. The second signal is sent to the wall of the mast cell, and it acts on arachidonic acid on the wall, and through two pathways, arachidonic acid will lead to formation of leukotrienes and prostaglandins. So during allergic asthmatic attack, the responses, as I mentioned, that it lead to degranulation of mast cell, and these asthmatic responses are divided into early and late responses. Early asthmatic response occur within minutes following exposure to the allergen, and involves histamine, leukotrienes, and prostaglandin liberation, and this will lead to bronchospasm and vasodilatation. The late asthmatic response that occurs hours later, and it involves secretion of cytokines, for example, interleukin number 4, interleukin number 5, interleukin number 16, tumor necrosis, factor alpha, and other mediators, for example, eosinophil chemoattractant factor, platelet activating factor, and neutrophil chemoattractant factor. So this will lead to cellular infiltration with blocking of bronchial lumen with mucus. Allergy test. Allergy tests are not used to diagnose asthma, but if the patient has allergies, that they may be triggering of asthma, so we can run allergy test just to identify the triggers for asthma and if the patient is allergic or not. So diagnosis, we use some methods, for example, skin prick test to determine the triggers or the allergens and to sort out if the patient is allergic or not. And this skin prick test include placing of a drop of allergen on forearm or back, and then prick it. In 15 to 20 minutes, wheals and flares will occur if the test is positive. Also, blood test. This may help to determine the type of allergen by running a test known as RAST, or radio allergen sorbent test, and this just to measure levels of specific IgE antibodies. Thank you very much, Professor Ahmed Bolan.